Hello everyone, I'm Byung-Cho from ExoCoBio. It's very nice to see all of you guys around the world. It's almost the end of December, so first of all, let me tell you, Merry Christmas. I wish you a Happy New Year. Okay, today my topic is about our rose stem cell exosomes and their clinical effect. Especially, I'm gonna focus on neocollagenesis. This year again, I have been traveling around the world to give my scientific lectures on exosomes. About more than 350,000 kilometers I have been running for. Please let me give you an important update on these 10 questions to ask any exosome innovator. So this year, I have been seeing a lot of new exosome companies which claim that they have the best quality of exosomes, and I found that many of them don't don't have real exosomes or real exosome manufacturing process or a quality control, that kind. So here, as of now, ExoCoBio is still the biggest investor in exosome science and manufacturing. And very fortunately, we have been seeing much more scientific publications from inside or outside, for example, our doctors around the world. So now we have 31 publications of clinical or non-clinical studies, and we are happy to expect 8 more publications in 2025. ExoCoBio has been committed to produce or to do this kind of scientific research or studies to help the patients or doctors around the world. And we now have 72, actually now it's 73, including 9 US patents. And most importantly, we have been generating the biggest number of clinical cases, especially here you can see that we are expecting 1.8 million clinical cases by the end of 2024 in more than 50 countries, which means that our product has been proved in terms of efficacy and safety. So this is my main topic for today. We have been spending about 4 years to characterize and isolate the best quality of rose stem cell exosomes. And many doctors are questioning how plant extracellular vesicles or plant-derived exosome can increase collagen in our skin. So we try to give some scientific publication and our data in terms of microRNAs or our proteomic analysis or our lipid analysis. But today, please let me focus on the biological functions of microRNAs contained in our rose stem cell exosomes. So this is the first publication I'd like to share as an important reference. And this publication is talking about cross-kingdom regulation by plant-derived microRNAs in mammalian systems. So in this publication, the authors uh, gave 4 examples for different type of plants or fruits. They identified 4 different types of microRNAs which can give an important biological function in our body. So here, for example, microRNA 168, they are targeting LDLRAP1, and they can do something in our cells. So as far as I know, there are about 35,000 exosome publications totally. In the meantime, only about 500 publications are about this kind of plant-derived exosomes or extracellular vesicles. So we need to understand that scientists are trying to find more exosomes or their biological functions these days. But definitely, scientists have been proving that there is a very important cross-kingdom talks between plant exosomes and human bodies or other animal systems. So this is the executive summary on my topic for today. ExoCoBio is the world's first company who developed and commercialized a new patented exosome technology based on rose stem cell derived exosome. Actually, ExoCoBio is the world's first company for any kind of plant stem cell derived exosomes or extracellular vesicles. We started this project about 6 years ago, and we have the biggest number of patents on this kind of new plant derived exosomes. And also, plant derived exosome can increase collagen synthesis in our uh, fibrodermoblast, especially for neocollagenesis. We could identify microRNA 574 and microRNA 2046. They can promote collagen synthesis, and very interestingly, those two microRNAs are two of the most abundant microRNAs contained in rose stem cell exosomes, which are shared by human stem cell exosomes. We are going to publish all the data in 2024, 25. In addition, my team found that another interesting microRNA named 130 is detected in our rose stem cell exosome, and also it was reported by a group of third party of scientists to promote collagenesis. And in our rose stem cell exosome, they are containing several important microRNAs as below. You can see here, LT7 is one of the most famous. It's a Nobel Prize 2024. And the function of LT7 is anti-inflammation and anti-cancer. And I found another publication on the biological function of microRNA 122. It can grow our hair. Also, there are three important microRNAs named 21, 23, 29. They are responsible for cellular proliferation or anti-scarring. And another microRNA named 125 is about anti-melanogenesis or anti-pigmentation. So as you can see in this executive summary, our rose stem cell exosomes can deliver these important microRNAs as a intercellular or cross-kingdom delivery. Actually, I use this slide for many congresses during my lectures, and scientists categorize many microRNAs into these three biological functions: anti-inflammation, cellular proliferation, migration, and angiogenesis, and the last is anti-scarring. As you can see here, LT7, 21, 23, 125, they are working for very important physiological functions in our skin cells or other types of cells. This is our data. My team was successful in isolating stem cells from rows, and when we culture those new stem cells. In the conditioned media, we could see that very nice new type of intercellular vesicles. And now we have been characterized some important ingredients in our RSCE, rose stem cell exosomes. And in terms of microRNAs, we found that 28 microRNAs are matched with human microRNAs. So again here you can see the many LT7 family of microRNAs 122, 130, 21, 23, 29, 125. And among them, the most or top 6 microRNAs from our root stem cell exosomes is the 574 and 1246. Let's talk about more details. So here, scientists discovered that microRNA 574 containing exosomes, they are delivering 574 for neocollagenesis. When we look at the key data here, you can see that they try to deliver microRNA 574 to certain kind of cells here, LX2, and that specific microRNA can increase the gene expression of collagen type 1A1. And, but in the meantime, inhibitors against microRNA 574 they couldn't increase. So this is very important microRNA that the effect of microRNA 574 can increase the expression of collagen type 1 and alpha SMA in our cells. In this data, they try to count the amount of collagen type 1 by the treatment of microRNA 574. So before and after, or with microRNA 574, the amount of collagen and the amount of alpha SMA were also increased together. And this is my second publication for share. You can see that very specific plant Veta vulgaris, they are releasing exosomes, and the scientists told that these new types of exosome can increase the production of collagen type 1 and 3 and hyaluron synthetase enzymes together. So when we look at the key data here, in a dose-dependent manner, these new type of exosomes can increase collagen type 1 expression and collagen type 3. It depends on the type of the collagen. This is short reference. You can see that scientists found that wheat-derived nanovesicles can promote in vitro wound healing activity. So in this publication, the key data is that with exosome, they increase the gene expression of collagen type 1A. So this is about the increased amount of mRNA of collagen type 1A gene, human donor fibroblast cells. Okay, this is the first reference. Here, growth fruit derived extracellular receptors can work for wound healing. So there are many data in this publication, but when we look at panel B and C, this grapefruit exosome increase the gene expression of wound healing related genes, collagen type 1, fibronectin, laminin, and vimentin. So you can see that collagen type 1 was also increased. In the panel C, they try to measure the expressed proteins of collagen type 1 and others. Here still you can see that collagen type 1, the amount of collagen type 1 was increased by the treatment of grapefruit extracellular vesicles. But in this paper, they didn't mention the exact number or types of microRNAs. Here, even one seed plant, like this one, Eclonia cava, and then they found that if we isolate from Eclonia cava, carry heat shock protein 70, and this protein can increase collagen. So you can find more data, but the key is that in aged cells by photoaging, when they treated this very special type of seed plant derived exosome, they could increase the amount of collagen 1 and 3. And here, this is about the effect of anti-photoaging collagens induced by microRNA 2046. And in this study, scientists manipulated to increase they try to overexpress microRNA 1246 in exosomes. So when we look at this panel B, overexpressed exosomes, I mean overexpressed exosomes containing microRNA 1246, they could increase the production of the protocollagen type 1. And when they applied the microRNA 1246 overexpressed exosome on the skin of mice, they could see very nice improvement in terms of skin regeneration or wound healing. And this is about the function of the microRNA 138. It's about scar, hypertrophic scar. So in these publications, scientists found that in hypertrophic scar, they could see more amount of microRNA 138, and they found when they treated microRNA 138 in human fibroblast, they could increase before and after or without with type 1 collagen and type 3 collagen, alpha SMA. They are all increased by the treatment of the microRNA 138. And in the meantime, this kind of microRNA 29B, they can reduce the amount of collagen too. So sometimes, or mostly microRNAs, important microRNAs we found, they can increase the amount of the collagen synthesis, but this interesting enzyme, this interesting microRNA, 29B, can decrease the collagen expression. And this is the summary. So microRNA 29B can reduce the collagen expression in fibroblastic state. And another microRNA contained in our raw stem cell exosome named microRNA 21, it can promote wound healing. By this pathway, microRNA 29, 21 can block STAT3, can increase TIM3, something like that. I hope you can read all the publications. So, maybe the first summary is that our raw stem cell exosomes are containing many different types of microRNAs, and some of them they can do neocollagenesis, for example, 574 or 1246. And this is about hair. I mentioned that microRNA 122 was also contained in our rose stem cell exosomes. And I could find an interesting publication. This is about human adipose stem cell exosomes. They are containing microRNA 122. They are antagonize the inhibitory effect of this hormone in hair follicles, which led to hair growth. So it's quite interesting that human adipose stem cell exosomes are carrying high amount of 122, and the same type of microRNAs are contained in large stem cell exosomes. And again here, this publication came in 2018. They are saying that LT7 regulate alpaca hair growth by downregulating ectodiplastin A. So key is that LT7B can grow longer hair in alpaca. So for with many slides, with many scientific publications I found last week, I try to share that important knowledge that which shows that all the publications are showing that microRNAs or important ingredients contained in rose stem cell exosome or plant stem cell exosomes, they can deliver biologically functional molecules to human cells. So the key is that microRNAs 12, 46, and 574, they can increase neocollagenesis in human donor fibroblasts. And very fortunately, ExoCoBio, actually, I was notified uh, by my patent lawyer in European countries. The European Patent Office granted the world's first plant stem cell derived exosomes as a patent for ExoCoBio. So you can see that claim one, we are the first to share this important scientific discovery on the cosmetic use of the exosome derived from rose stem cell exosome as an active ingredient for skin regeneration, elasticity, and wrinkle reduction. We are very proud to be granted with this world first patent from the European Patent Office. Please remember this executive summary again. Thank you so much. ExoCoBio is the world's best quality exosome producer. We are second to none. We are committed to provide or to lead this exosome industry. Thank you so much.